Hello everybody, this is Akshay over here and welcome to my another video, and in this video we'll be discussing about biological classification chapter 2. So Aristotle was the first person who actually made an attempt to classify the organisms. Like he tried to classify the organisms such as plants into tree, shrubs and herbs based upon their simple morphological characteristics. He even tried to classify the animals on the basis of their blood color, that is, organisms having red blood color into one group and those who didn't have the red blood color into another group. Later, during the time of Linnaeus, there was two kingdom classification, that is Plantae and Animalia. But the problem with this two kingdom classification is that it did not distinguish the unicellular and multicellular, prokaryotic and eukaryotic organisms, photosynthetic and non-photosynthetic organisms. So to overcome this problem, R.H. Whittaker introduced a five kingdom classification, and the five kingdoms are Monera, Protista, Fungi, Plantae and Animalia. So what were the criteria that were considered for the five kingdom classification? In fact, it is cell structure, thallus organization, mode of nutrition, reproduction and phylogenetic relationships were considered for the formulation of five kingdom classification. So prior to the five kingdom classification, the bacteria, blue-green algae, fungi, gymnosperms and angiosperms were actually put into Plantae. The reason was all of them actually had cell wall. Unicellular and also multicellular were also present in the same group. But later, after the five kingdom classification, prokaryotes were placed into Monera, eukaryotes were placed in Protista, and fungi got its own kingdom also. So let us begin with each of these kingdoms. The first kingdom is the Monera. So the main representatives of Monera are the bacteria. They also show very diverse shapes. If it is a spherical, then we refer it as coccus. Rod-shaped, we refer it as bacilli. If it is comma-shaped, then it is called as vibrium. And if it is a spiral, then it is called as the spirulum. They also have the highest diversity of metabolic activity or metabolic diversity. The mode of nutrition, some of them are actually autotrophs, but majority of them are actually heterotrophs. One representative of Monera is Archaebacteria. They are primarily different from the bacteria based upon their cell wall composition. They are mainly found in harsh habitat conditions. For example, if they are mainly found in salty areas, then we refer them as halophytes. If they are mainly found in hot springs, then we refer them as thermoacidophils. If they are found in marshy areas, then we refer them as the methanogens. In fact, methanogens are the agents which are present in the gut of the ruminants, and they are responsible for production of methane gas. Now, eubacteria, some of them may be photosynthetic, such as the blue-green algae. They are colonial, or they may be unicellular, or they may be filamentous. Now, Anabaena and nostoc do have the nitrogen fixation ability, and they can do this because of their having special kind of cells called as the heterocysts. Now, some bacteria are chemosynthetic autotrophic in nature, means they can oxidize nitrates and ammonia to produce the ATP. Some bacteria are also heterotrophic in nature. They are involved in curd making, nitrogen fixation, and also they can cause diseases. Some of them also cause diseases. The Mycoplasma is known to be the smallest living being. It has no cell wall and they are anaerobic in nature. Protista. It mainly includes unicellular eukaryotes. They provide a link to plants, animals and fungi. So chrysophytes, one representative of Protista. Examples mainly include diatoms and golden algae. So diatoms have overlapping cell walls and they are mainly embedded in silica. They are the chief producers in the oceans, and throughout the course of time, diatoms have been deposited into the oceans, and it is referred as the diatomaceous earth, which is mainly used for polishing and filtering syrups and oils. Next is dinoflagellates. They have cell wall, but it is made up of cellulose, and they have two flagella. One flagella is longitudinal, another flagella is a transverse, but it is embedded into a furrow between the cell plates or cell wall. Now, red dinoflagellate gonolix cause reditypes. It is mainly the rapid multiplication that leads to poisoning of the water because of release of toxins. As a result, many marine organisms die. The next is euglenoids. They are mainly found in the stagnant fresh water. They do have a protein coat called as pellicle, but they do not have cell wall. As a result, their body is very much flexible. Euglenoids can be both photosynthetic or heterotrophic in nature, depending upon the presence or absence of sunlight. If there is sunlight, then they are photosynthetic. When there is no sunlight, they become heterotrophic in nature. Now, slime molds, they are primarily saprophytic in nature. Under certain conditions, they aggregate to form plasmodium, and later they differentiate to fruiting bodies. The fruiting bodies mainly contain the spores, and the spores are dispersed by the air. Protozoans. There are primarily four types of protozoans, that is amoeboid, flagellated, ciliated, and sporozoans. Example for amoeboid protozoan is amoeba. It is having pseudopodia for locomotion and capturing of food. Flagellated, an example is the trypanosoma, which causes sleeping sickness. Ciliated protozoan example is paramecium, and an example for sporozoan is plasmodium, which causes malaria. Next is kingdom fungi. Now, in the case of fungi, the cell wall is made up of chitin, and the mode of nutrition is heterotrophic in nature. So the body is mainly made up of elongated thread-like structures called as the hyphae, and the network of hyphae is called as the mycelium. The hyphae are of two types. It is a cenocytic or septic. So the reproduction is mainly by vegetative means, that is by fragmentation, fission, or budding. It may be even through by production of asexual spores such as conidia, aplanospores, and zoospores. And if it is sexual mode of reproduction, then it is oospore, ascospore, or basidiospores. The steps which are involved in the sexual reproduction are as follows. Plasmogamy, that is fusion of protoplasm. Karyogamy, fusion of the nucleus. And later, in the zygote, which is resultant of karyogamy, there is a meiosis. And after meiosis, there is haploid spore formation. In some cases, such as ascomycetes and basidiomycetes, there is an intervening stage called as the dikaryon phase. Means inside a single cell, there are two nuclei. The classification of fungi is as follows. Phycomycetes, ascomycetes, deuteromycetes, and basidiomycetes. In the case of Phycomycetes, the mycelium is cenocytic and it is aseptate. The asexual spores are zoospores, which are motile in nature, or aplanospores that are non-motile in nature. And these spores are endogenously produced. Examples mainly include mucor, rhizopus, and albuco. Albuco is a mustard plant parasite. The next one we are having is the ascomycetes, also called as the sac fungi. The mycelium is branched and septate. The asexual spores that are produced are conidia by a structure called as the conidiophore, and they actually germinate to form the mycelium. The conidia germinates to form the mycelium. The sexual spores are produced by structures called as the asci and are called as the ascospores, which are present on fruiting bodies called as the ascocarp. Examples mainly include claviceps, neurospora, aspergillus, penicillium, and yeast. Yeast is unicellular fungi. Neurospora is mainly used for genetic and biochemical studies. The next one is the deuteromycetes. The mycelium is branched and aseptate. Only asexual or vegetative phase are actually recorded or observed, and hence deuteromycetes is called as the imperfect fungi. Asexual spores that are produced are the conidia. They are primarily decomposers, and examples are alternaria, calectotrichum, and trichoderma. The last one is the basidiomycetes. The mycelium is branched and septate. Asexual spores are not generally found, but fragmentation is most common method of reproduction, that is vegetative propagation. Examples mainly include agaricus, smut, that is the pistilago, and puccinia. It causes wheat rust disease. So the next is viruses, viroids, and lichens. The term virus was coined by Pasteur. Ivanovsky observed that there were some agents which were smaller than bacteria that was causing disease to tobacco. Later, Bijernik, through his experiment, observed that a healthy tobacco plant can be infected with an extract of an infected plant. And the result was infection in the healthy tobacco plant. So he called this extract, that is the fluid of an infected plant, as a contagium vivum fluidum, which means infectious living fluid. Later, Stanley crystallized viruses. So crystallization of virus mainly showed the structure of virus is mainly made up of a protein coat called as the capsids, and the units that are involved in the formation of capsids are called as the capsomeres. The virus also have genetic material. Their genetic material may be either DNA or RNA. They cause disease in various organisms. Viruses that cause disease in the case of plant, they have single stranded RNA as genetic material, but the viruses that cause disease in animals, they have single stranded or double stranded RNA as genetic material, or simply double stranded DNA as genetic material. Later, Theo Diner discovered a unique disease causing agent, that is the viroid, which caused potato spindle tuber disease, which was only RNA. It had no protein coat, and also it had very less molecular weight. So the last one is the lichens. Lichens are mainly symbiotic association, means it is not an individual, it is actually made up of two individuals, that is the algae and the fungi. The phycobiont is called as the algae, mycobiont is called as the fungi. So they mutually help each other for the nutrition and growth. So if you found this video very helpful to your preparation, then do like the video and share it among your friends, and finally subscribe to this particular channel. Thank you.